Okay, I guess we're live. As usual, if you can hear me and if you can see my screen and everything else, just let me know so that I know things are working. Just drop a comment in the chat and I'll be sending out some invites so that people can join. Announcements. Why the fuck did this thing did not work? Um. Okay. Let's see. I'm getting some warning from YouTube, so let me know if things are working. All right. So, this is a topic that I wanted to make a video about or stream about for a long time. Actually, I've been wanting to do like a video, but I got too lazy. And the reason this interest was kind of rekindled was that there was a recent event in India, which is that in West Bengal, which is like an eastern state in India, the government put out like um, they put out like a tender for a contract or something like that for serving midday meals in government schools. The idea of a midday meal is that you offer some meals to kids, and that would, motivate uh, the parents to send their kids to school so that they don't have to feed the kid for at least like one meal a day or something like that. Okay, and that would incentivize people to send their kids, and thus the kids get educated. That is kind of the motivation for these midday meals. Now, obviously, if a government is providing midday meals, it is expected that they provide, um, firstly, the meal should be nutritionally complete and like balanced or whatever. Um, and also, in my view, just as important is that the meal should be hygienically prepared. And this is something that's a super like like we all know about the videos that come out on like YouTube or whatever about how certain things are prepared in India. And a lot of these things are not very hygienic, and it's important that the food that the kids get is hygienic. Now, as part of that whole thing, there were people who were invited probably to bid to win the contract. And there is this religious Hindu organization called ISKCON, which is the International Society for Krishna Consciousness. A lot of white people um, have embraced Hinduism as a result of the activities of this organization. It's a pretty spread out organization, even internationally. And the headquarters of that happens to be in West Bengal, because the person who was the founder, Srila Prabhupada, was a Bengali guy, and the headquarters are in Bengal. So a lot of white people, you can actually, like, if you go to like Kolkata airport, which I did a few months ago, I could see plenty of white people, like plenty in the sense maybe like 5 or 6 white people who were adorned in like Hindu clothes, in the sense that they had the Hindu haircut, which is like most of the head shaved and just a lock of hair um, that's long, and they had like beads on their neck, and they were wearing like um, you know white cotton clothes or something like that. It's kind of like a the Hindu way of dressing, and I'm assuming that that is like people who have converted to Hinduism as a result of, well, maybe they've converted, maybe their parents converted and raised them into the faith or whatever. I don't know exactly, but plenty of people convert to Hinduism because of ISKCON, and they have this. So ISKCON uh, placed a bid in that contract to get that contract, and obviously they are a non-profit organization, so they took a cut. They took a hit themselves to make the cost affordable or something like that, and they won the contract. However, the so since they won the contract, they will now be providing midday meals. However, ISKCON is an organization that promotes vegetarianism pretty heavily, right? So they said that we're not going to be providing eggs to the kids as part of the midday meals, and that ruffled a lot of feathers. This is like a huge issue on Twitter. Everyone was having like a meltdown on Twitter about, oh my god, like these kids are going to be deprived of nutrition or something like that, which is crazy. Like, I don't know why they would think that, but um, and this came even after the government issued clarifications that standards have been set for nutritional completeness, and it was made clear that even though eggs are not going to be provided, the protein requirements of the kids are going to be met with other sources like soy protein, um, um, like kidney beans and chickpeas, and a bunch of other things, which includes cottage cheese. So these kids are going to be provided with all these different sources of protein for their meals every day, instead of eggs that they. I'm not really sure. I'm not sure what was the frequency of eggs. I'm not sure if they got eggs every day or if they got eggs once a week or twice a week or something like that. I don't remember exactly. But the point is that back in the day, the food was not super hygienic. It was, made by whoever, and like there were lots of reports that the food is just extremely unpalatable, and like you wouldn't feed that to your worst enemy. And in that sense, ISKCON obviously provides a much better option, in the sense that they are going to be providing food that is pretty like palatable, and on top of it, they are going to be ensuring that eggs are not going to be provided, and that will be replaced with other plant-based protein sources, and also dairy, which we'll talk about later, I guess. Um, but one common theme that happened during that discussion was that everyone was jumping on this bandwagon to um, yeah, everyone was like jumping on the bandwagon to comment about this and be like, well, they're going to be given all of these protein sources, these other like plant-based protein sources, but they're not bioavailable. And you can tell that these people are not super literate when it comes to nutrition. They have no idea what they're talking about when they say um, protein is is not bioavailable or something like that. So I thought that there should be like a video that is made. I'm sure there are other resources about this, but I think there should be a video that explains the underlying concepts, um, and explain the concepts that are adjacent to veganism, um, not just protein in general, but also like since this is like a thing that's adjacent to like plant-based diets and something like that, there has to be a discussion which goes in that direction. So I've decided to make this video where I will be going through a bunch of um, facts about protein and like just how bioavailability and all these concepts tie in together. So I'm just gonna take a quick look at the comments before I proceed. We should also keep in mind that kids below the age of 13 require less than 34 grams of protein every day. Yeah, I mean this is going to depend on the body weight. Um, and obviously like it's not hard even if we exclude soy chunks. Yeah, sure, but the point is, why should we even think about excluding soy chunks? Anyway, but it's a separate topic altogether, so I will just go through this. So just an introduction. When we say protein, there are some standard definitions of the term protein, and there are some related terms. Um, Obviously, there's let me use a different color. There is a term called amino acid. There is a term called polypeptide. Sorry, I should say polypeptide. These are the kinds of terms you will encounter when you're talking about protein. So let's just define what a protein is. A protein is a chain of amino acids. Now, roughly, that is going to look like that. So that is like a standard form of an amino acid. Um, so you have, oh, sorry, 1 second. I drew it wrong. Yes. So you have over here, that's a standard form of an amino acid. So an amino acid is called amino and acid. The amino part refers to this group, which is the nitrogen along with 2 hydrogens. That's the amino part. And the acid part is this part over here. Okay? So this is a carboxylic acid. Over here, this interjection is like a C, which is double bonded to an oxygen. And there is a hydroxyl group over here, and this is called a carboxylic acid. And that's why this whole structure is called an amino acid. Now, one point of confusion here should be left behind, is that amino acids are acids. Well, they are acids in the sense that they contain a carboxylic acid group, but they don't. Not all amino acids have acidic properties. When you're talking about your acids and bases, there are things that have acidic properties, and there are things that have basic properties. And like the idea is that there has to be like a group that like donates protons. If it donates protons, then it's an acid, and if it receives protons, then it is a base. That's the kind of thing. So in the sense that, like if you have hydrochloric acid, which is HCl, when it dissociates, it's going to dissociate into H plus ions, which is the same thing as a proton, because a hydrogen atom is composed of a single proton and a single single electron, and if you lose the electron, that's how you get a positive charge, and that's how you have a proton. But without without an electron, it's just a proton. So if the concentration of hydrogen ions increases, the thing is more acidic. Blah blah blah. That's some adjacent chemistry that I don't want to get into. But yes, amino acids, because there are like there are a bunch of amino acids. There are like about 20 of them. Sometimes you can consider like a little more than that, but standardly you can consider like 20 or 21 amino acids, and they're not all acidic, which is why this is like a point of confusion that needs to be left behind right at the start. All right, so polypeptides. Why is it called what's a polypeptide? Poly means many, and peptides means so there's a bond in the amino acid which is called a peptide bond, and that's why when you have chains of amino acids, it's called a polypeptide. So you have, let's say, methionine, and then leucine, and then histidine, and then lysine. Sorry. And they're all connected to each other. This would be like a polypeptide. So a protein is essentially a polypeptide. And there can be different permutations and combinations of what constitutes a like which protein it is. There can be different proteins which have different amino acid sequences, and that amino acid sequence is what distinguishes each of these proteins from one another. Okay. Now, let me go over here. So, I hope y'all can see this. Yeah. So, as I mentioned, it's a chain of amino acids, and there are, as I said, there are 20 amino acids, and the thing that, that differentiates proteins from one another, amino acids from one another, is this R group, which is called an alkyl group. So you can have something like this. This is the simplest amino acid, called glycine. And if I do essentially the same structure but I change just one thing over here. I retain this hydrogen, but I change this to a CH3. This is a completely different amino acid, which is alanine, right? So this R group over here is what separates different amino acids from each other. So you have glycine, you have alanine, there's a bunch of others which I've listed over here. Some of the 20 amino acids are essential amino acids, and they're called essential because they're not synthesized by the body and they must be obtained from diet. That's why they're essential, because we lack the biochemical pathways that are necessary to synthesize these amino acids, so we just obtain them from our diets. And histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, valine. These are the amino acids which are considered essential. Some people mistake creatine to be an amino acid. Creatine is not exactly an amino acid, but that's a completely different thing. Anyway, now let's move on to the meaning of the term bioavailability. This is something that is very flippantly used, and people don't really know how to interpret the term. They say that different things have different bioavailabilities without actually knowing what it means. So I've defined this like a property of a food that relates to how well its protein content is digested and absorbed by the body. So the degree to which a particular protein or a particular the protein from a particular food is digested and absorbed by the body is what I call bioavailability. And there's a related concept called protein quality. Now, a lot of people, when they use these terms, they mix up bioavailability and protein quality. What they're referring to is protein quality, even though they use the term bioavailability. And protein quality is the property of food that relates to how much of our protein requirements are met by consuming this food. And requirements are again defined by our norms with respect to protein consumption. Now, there have been some attempts to sort of formalize this concept of protein quality. And one of the, normie kind of ways is to assess the completeness of the protein. Now, there may be certain foods. Let me just draw like a bar graph. So let's say on the x-axis you have amino acids, and on the y-axis you have the amount of protein in, say, 1 gram. Amount of that amino acid in 1 gram. Okay, so there can be some proteins which have like a really high amount of certain amino acids, and basically none of other amino acids. Okay, again, even here there's like two senses of completeness, in the sense that a particular protein source contains all the amino acids, which is that the protein should contain all essential amino acids. Now, there are very few proteins in nature that lack all the amino acids. If I remember correctly, is it collagen? I think collagen is the one that lacks one amino acid. Don't quote me on that. But there are very few amino acids very few protein sources in nature that lack any amino acid. completely, right? So there's always a different amount of amino acid present in these sources, right? The other sense is that you have high amounts of some particular amino acid, but you have lower amounts of the other essential amino acids. And that's what we kind of mean by essential. What we mean by completeness. So, but nobody cares about it, because we have developed other metrics that have tried to like formalize what we mean by protein quality. Okay. The first one that we are going to discuss is the protein digestibility corrected amino acid score, which is roughly the ratio of digestible limiting amino acid in sample versus reference. And the reference is the theoretical ideal, usually egg or whey. And this is expressed as a percentage. Now let me unpack all of that. There's a bunch going on here, okay? So this is the formula for PDCAS. The number of milligrams of limiting amino acid in 1 gram of test protein. What does limiting amino acid mean? Limiting amino acid is the amino acid that is present in the smallest amounts relative to other essential amino acids in 1 gram of test protein. So that's the limiting amino acid. So for example, in wheat protein, for example, or seitan, for example, it has low amounts of lysine. So lysine over there is the limiting amino acid. Which is the amino acid that's present in the smallest amount compared to all other amino acids in 1 gram of test protein. And that divided by the number of milligrams of that same amino acid in 1 gram of the reference protein, which is, let's say, egg protein or whey protein. And this whole thing is multiplied by the fecal true digestibility times 100. Okay, so what is fecal true digestibility? Now, the fecal true digestibility is the amount of protein that is, so let's say, let's say this is like the mouth, and there's there's the stomach, and it goes into like your intestines, and then it goes into your large intestine, and then it comes out through your colon, right? Now, if I consume certain amount of protein, I know how much of that protein is entering the body. I can compare that amount that I put into my body with the amount of protein that I am left with from a person's poop, right? So you compare the amount of nitrogen that is in the stuff that they put in their mouth, and you measure the amount of nitrogen in the a person's poop, so you know how much of it has been left inside the body, which means it has been absorbed by the body. So that is the factor that is over here. One second. All right. So that's this fecal true digestibility. Okay. Now there has been a second attempt, which is supposed to be an update to the PDCAS, which is the DIAS score, which is the digestible indispensable amino acid score. Now, indispensable amino acids kind of the same thing as essential amino acid, and the formula for DIAS is 100 times the ratio of the amount of digestible dietary indispensable amino acid in 1 gram of dietary test protein, and the amount of the same amino acid in 1 gram of reference protein. So let's kind of understand what that means. It means over here, oh, by the way, there's a difference in what digestible means in the DIAS score versus the PDCAS score. And the difference is that the PDCAS score uses fecal to digestibility, which I've already explained, and the fecal to digestibility was estimated, if I'm not wrong, by feeding rats. Okay. The DIAS score improves upon it by 2 methods. Firstly, there are some limitations with this particular way of measuring absorption, because the nitrogen that you put in your, like if your food contains certain amounts of protein or nitrogen, your body is obviously going to absorb some of it. Your body is going to absorb some of it. However, the stuff that is not absorbed by your body is going to be pushed out in your feces. Now, if we remember that there are a bunch of microorganisms that are in our guts, right? So there are certain bacteria, there are certain other organisms, microorganisms, that are present in our colon and our large intestine, such that they may use some of that protein, some of that nitrogen in the feces for their own purpose. So there's some amount that you put in your body. So let's say there's a total protein, right? So the total protein is going to be the amount that you wait 1 second. What am I thinking? The total protein is the amount you put in your mouth, right? That which is the entire thing. Now that is going to be an addition of the amount that was absorbed by your body, plus the, let's say, nitrogen. Let me just put this as nitrogen absorbed by body, plus nitrogen used by bacteria, or let's say microbiome, plus nitrogen in the feces, right? Now, what the fecal digestibility ignores is this part. It considers these two to be the same thing as this, because it's assuming wait 1 second, I think I misspoke over there. So it considers that it ignores the the nitrogen that is used up by the microbiome, which means that it is kind of overestimating the amount of protein that is absorbed by the body, because then you're going to be like, there's some amount which is if you say this is 100, okay, and then you say this is 50 plus 25 plus 25, but if you ignore this and you measure 25 over here, then you when you're trying to find out how much is absorbed, you will kind of, and if you ignore this, it means that this is 0, right? So your calculation will end up showing that N absorbed is actually 75 and not 50, which it actually is. So there's an overestimation of the protein that is absorbed, which is why PDCAAS scores are generally better for the same protein compared to DIAS. Now, the other thing that's wrong with PDCAAS is that PDCAAS caps all scores to like 100%. So it's expressed as a percentage. So it caps the score at 100%, or if you prefer if you prefer to keep your score from 0 to 1, then you can keep it 0 to 1. But it's like, you can also interpret it in terms of percentage, so that'll be 0 to 100, right? Whichever way you prefer to use it, it caps the score at 100%. So what you're losing, what happens is you're losing the differences between proteins that are both 100%. So let's say whey protein is 100, and egg protein may also be 100, but one may be absorbed more than the other. You just don't know, right? So it caps the score at 100. Thus, it makes the comparisons at the top much more difficult. Now, DIAS is gets rid of that by not restricting the score to 100. So you can have DIAS scores more than 100%. And that will tell you some proteins which are more than 100% and some proteins which are less than 100%. Now, the way you interpret the DIAS score is that if a protein has a score of 100, that means that if you consume the estimated average requirement of protein, which is 0.66 grams per kg of body weight per day, that will satisfy 100% of your requirements. That's how you interpret the DIAS score. However, I have problems with this number, which is the 0.66 grams per kg as being the EAR, which we will get to in just a second. So that's how you interpret. So if a food has a DIAS score of 90%, for example, or 0.9, then you can assume that if you only consume that food such that you met your estimated average requirement, then you would meet 90% of your protein requirements. Okay. Now, yes, did we talk about? Yeah, so PDCAAS has some limitations, which is that it, as I mentioned already, there may be some protein which is not absorbed by the body, but that protein is used by the microbiome, which is after the ileum. The ileum is the end of the small intestine. Wait, one second. No. There's duodenum, there's jejunum, and ileum. Right. So, all the protein that is absorbed after the ileum should not be considered, because all the protein is absorbed that is supposed to be absorbed by our body is absorbed in the small intestine. So anything that is not absorbed in the small intestine is not going to be absorbed in the large intestine. So whatever happens in the large intestine, whatever reduction happens in the large intestine is because of the microbiome. And that leads to an overestimation of absorption, as I showed you already. And it truncates the values at 1 and leads to loss of comparison between all the proteins that rank number 1. Okay. Now, there are limitations with DIAS as well. It's not a perfect score. One of the biggest limitations is that the effect of food preparation is not studied there. Is not studied. Also, the estimation of the ileal digestibility, which is, you have someone sticking like a needle into the abdomen to figure out how much protein is in how much protein is found at the end of the small intestine, which gives us a reasonable estimate of the absorption. Now, that study has been done in pigs, so we're still not doing these estimations at the human level. The DIAS score assumes absorption in pigs are the same as absorption in humans. That's one of the biggest flaws. Even though pigs and humans are pretty close, they're not the same. Okay. And it studies it ignores the food preparation, such that like sometimes you cook certain foods to a certain temperature, you boil some foods, you roast some foods. There are a bunch of there are some foods that are cooked in the presence of high amounts of salt. So there are a bunch of different ways that we process foods, and the preparation of foods affects the amount of protein that is absorbed, but that is not accounted for in the DIAS. The second thing is DIAS focuses only on atomic food items as opposed to evaluating composite foods. Now, what does that mean? It means that, for instance, there can be foods that are, like dal and rice is like a combination. So there are some, and like rice is never really eaten alone. It's always eaten along with dal and or some other kind of thing, right? Or like it's eaten with like rajma or chole or something like that. So rice is seldom eaten by itself, unless it's some like some insane like rice only diet or something like that, which is not something I would encourage people to consume. But rice is not something that's consumed on its own. So it's like when you rate foods individually, you kind of penalize them, even though they are not going to have a detrimental effect in virtue of missing some amino acids, because those amino acids are recoverable from the foods that they're usually paired with. Okay. So that's the limitation, that it focuses on single foods as opposed to a composite dish. Thirdly, again, this one ties into the previous point, which is that individual foods can have low DIAS score, but still be decent sources of protein, given that they have decent protein to calorie ratio, etc. Which is that you can have a low DIAS score, but if the protein per calorie ratio is high, then you can still consume that protein to a high enough amounts and recover as much you can get. Now, none of this means that plant protein is necessarily inferior to animal protein in terms of hard outcomes. It doesn't necessarily mean that vegans have to eat more protein, mainly because DIAS scores are additive. And for people who are undergoing resistance training, as long as you're consuming 1.6 grams per kg of body weight per day, and you're consuming 10 grams of leucine, so whatever protein you're consuming must be 1.6 grams of protein per kilogram of your body weight per day, and have 10 grams of leucine. As long as those two criteria are met and you're training sufficiently, there is no difference in strength or lean body mass gains. Lean body mass is, like your muscles, etc. Anything that is not fat. So there's no difference in strength or LBM as long as people consume 1.6 grams of protein per day with 10 grams of leucine. Now this comes from this study, which directly measured the impact in people consuming whole food plant-based diets. Oh wait, I'm not sure if they're consuming whole food. I don't forget about that. They're consuming a plant-based diet, which is high protein, and the protein amount is matched to the participants in the omnivorous group, and they're given the same amount of resistance training, and that's how they figured out that as long as you consume adequate protein and leucine, there's no difference in the strength or lean body mass. Now, why is leucine important? Because leucine is the amino acid that saturates the mTOR complex, which is a pathway for, cell growth. So, as long as you have enough leucine, you are saturating your mTOR pathway such that you are maximizing your anabolic response, and thus there's no difference in strength or lean body mass. Now, for people who do not work out, who do not undergo resistance training, and who have food access, eating 1.2 grams of protein per kg of body weight per day is sufficient. Now, this is in contrast to what is said over here about the estimated average requirement being 0.66 grams of 6 grams of protein per kg of body weight per day, which is the estimated average requirement. So let me just take a step back and explain the differences between EAR and RDA. EAR is the estimated average requirement, which means it is the amount that has to be consumed such that 50% of the population is having their protein needs met. So if you consume that's the amount that's the average estimated average requirement. And the RDA is the amount for the vast majority, so about 98% of people. Okay, so when you look at protein recommendations, you will see 0.66 to 0.8 grams or something like that. So so this 0.66 is the EAR and 0.8 is the RDA. So if you want to be really sure that you are not under consuming protein, you should be hitting this amount, which is the 0.8. However, this is based on a kind of study called nitrogen balance studies. Okay, which is similar to what I said earlier. You can take some amount of nitrogen that is eaten, and then you can measure the amount of nitrogen that is excreted. If you know, when in our urine, urine contains urea, and urea is a nitrogen containing substance, right? So this is for us, like we excrete urea. If you go down to the underwater environment, over there the animals excrete ammonia, not urea. And birds will excrete uric acid. Okay, so these are the ways in which we excrete nitrogen from our body. And the idea is that if you are consuming sufficient amounts of nitrogen, then your stores in your body are going to be maintained. However, if you consume less nitrogen, then some of your muscle and other stuff is going to be broken down, and thus your proteins are going to be broken down. So let's consider two scenarios. You're deficient in protein, right? So if you're deficient, then some of your amino acids, some of your proteins are going to be broken down, because your body needs proteins for other more important things. So stuff from your muscles are going to be broken down, and that's going to be channeled back to other things. So overall, what's going to happen is there's going to be a metabolism of proteins, such that there is an increase in your nitrogen excretion. So there's going to be more nitrogen excretion, right? On the other side, if you consume enough protein, then the nitrogen is going to be retained, and your excretion of nitrogen is going to be lower. So that's how a nitrogen balance study is conducted, and that's basically how the protein requirements were being estimated until now. So they would just plot a graph, and it would be like, wait, let me just use this. They plot a graph, and it'll be like low amounts of protein, which means more excretion, and as the there's going to be like some kind of like decrease like this as the protein amount increases. Okay. And they just model this, and they figure out which is like what's the halfway point, which is going to be your EAR. So if this is the halfway point, then that's going to be the EAR, and the 98th percentile, which is going to be the is going to be your RDA, right? Now, there is a study that did a meta-analysis of many nitrogen balance studies, and instead of using a single linear regression, they used a two-phase linear regression. Now the thing is, typical dose response curves in biology and nutrition, it's more appropriate to use a two-phase linear regression, such that you identify like a breakpoint. So let's say if this is the curve, you identify this as one breakpoint, and identify this as another breakpoint. So this is the first regression, and this is the second regression, and the point at which you converge, you can consider that as the point at which requirements are met. Okay. Now, there's a study that conducted this exact experiment, and they over here, so this is a study, and over here they conducted the thing that I just mentioned, a two-phase linear regression, and they found that the estimated average requirement is 0.91 grams, and the RDA is 0.99 grams per kg of body weight per day, as opposed to the previous recommendations of 0.6 to 0.8. Now that's a 0.3 gram difference, right? Which means we are probably underestimating how much protein we should be consuming, even if we consider the nitrogen balance studies. So the two-phase linear regression is giving us a higher value for the EAR. But there's another method. So this is the nitrogen balance studies, which are kind of hard to conduct and measure accurately. So there's another methodology for measuring this, which is the indicator amino acid oxidation method. Now, what's that? Okay. So let's say you consume some amount of protein and you have a bunch of amino acids as a result of that protein being broken down into individual amino acids, right? Now, let's say there are some amino acids which are present in less amounts than other amino acids, which is, again, the limiting amino acid that we talked about before. And that limiting amino acid will halt the protein translation. So the way things work is you have a DNA. Like, protein synthesis is happening in every cell of your body right now. Like, it's happening, right? So the way it works is there's DNA, which provides a blueprint for things called mRNA, or just RNA. Like, RNA has different types. So DNA is the blueprint for RNA, and RNA is the blueprint for proteins, right? This is what we call the central dogma of life, that DNA, well, let's not get into that. The idea is that you cannot go from proteins back to DNA or whatever, because there's an information loss, whatever. We're not getting into that. The idea is there's a flow of information from your DNA. So there's DNA in every cell of your body, and that acts as a blueprint to make RNA. It's sort of like a miniature copy of a DNA. So you can consider DNA as like the blueprint of a car. Everything that you need to build a car, DNA is like a blueprint of that. And RNA is kind of like the blueprint for how to make the engine. And then there are factories in cells called ribosomes that read the mRNA and they make proteins, right? So ribosomes are the protein factories that are responsible for synthesis of proteins, and they require this RNA as an instruction sheet to make proteins. And different proteins have different instruction sheets or corresponding mRNAs. And the idea is, let's say there's an mRNA, and there is a ribosome sitting on this mRNA. As the ribosome proceeds, right, it recruits different transfer RNAs, and these transfer RNAs, they have different amino acids on them. So you can have a transfer RNA with methionine, another transfer RNA with lysine. Similarly, you have 20 different 20 different tRNAs for 20 different amino acids. Now, each of these tRNAs are going to be called into the ribosome, and they're going to associate with the mRNA, and then there are going to be some enzymatic activities that are going to transfer this amino acid from the first tRNA to the growing protein chain. So there's going to be a chain of proteins that is going to grow by transferring the amino acid from the tRNA onto the growing polypeptide chain. Okay. Now, let's say you have less amounts of one particular amino acid, less tRNA corresponding to a particular amino acid, which means there's going to be more time required for the ribosome to complete the same. Let's see if I can think of like an analogy. Let's say you want to make let's say you want to make a steering wheel. Okay, so you have let's say steering wheels are made of plastic, and there are some nuts and bolts that hold the steering wheel into place. The number of steering wheels that you can make in a given time in a factory depends on the availability of the raw materials. So suppose you have lots of plastic, but you don't have nuts and bolts, which are required to hold the plastic together to make the steering wheel. If you don't have the nuts and bolts, then you have to wait longer for each steering wheel to be produced, because you're waiting for the nuts and bolts to be available. And they're going to be available at a lower rate if the raw materials are not abundant. Now, if the raw materials are abundant, then you're going to have plenty of nuts and bolts, and you're going to have plenty of plastic, so you can make plenty of steering wheels in the same amount of time. So similarly, ribosomes get stalled, like the rate of protein synthesis goes down if there is a deficiency of certain amino acids. Okay, now what happens is, during that time, the other amino acids, they get oxidized, right? And what you can do is you can exploit this information to design an experiment. And what this technique does is it uses a C13 stable isotope, which is inserted into an amino acid called phenylalanine. So phenylalanine contains not all phenylalanine. There's a synthetically made phenylalanine which contains C13 carbon, which is a stable isotope of carbon. It's not carbon like a natural carbon. Natural carbon is C12. This is C13, which is heavier, right? And what happens is you can so if your body is deficient, again, you have two possibilities here. If your body is deficient in protein, the amount of phenylalanine that is going to be oxidized is going to be more. And the consequence of that is you release CO2, and some amount of that CO2 is going to contain the stable isotope carbon, right? Because the oxidized thing is eventually converted to carbon dioxide and then excreted from our air, from expired air, the air that you breathe out, right? Now, if you're deficient, more of this is going to be oxidized, and there will be more C13 in the carbon dioxide that you breathe out. Now, if you are sufficient, then the amount of C13 phenylalanine that is oxidized is going to be reduced. So there's going to be a reduced amount of C13 CO2 that is expired through our lungs, right? So you give someone a certain amount of protein, and then you measure the amount of C13 in their air, and then you increase the amount of protein over over different servings, and then you measure more of that. You keep making more measurements of CO2. So if the amount of C13 CO2 decreases, it means that you're achieving sufficiency. You're getting closer and closer to sufficiency. So there's going to be a graph that looks like this. So at low protein intakes, if your protein intake is low, then your indicator amino acid oxidation is going to be high. So it's going to be like this. It's going to fall over time. Not over time, over increasing amounts of protein. And they have done another two-phase regression, and they have identified like a breakpoint, and that breakpoint will be sorry, my drawing is a little horrendous. So if say that is a breakpoint, then that protein amount will be considered the amount that is sufficient, you know. So this is the amount that will be considered sufficient, because that's the amount where there is a reduction in the indicator amino acid oxidation. Sorry. Anyway, let's go back to this. And what they have done is they have considered that there are problems with the nitrogen balance method, and they have taken the alternative indicator amino acid technique, and they have determined that the requirements are more. So the requirements that are estimated are 0.93 to 1.2, which is why in my recommendations over here I said that eating 1.2 grams of protein per kg of body weight per day is sufficient for someone who is not training, who's not involved in resistance training. So that's the theoretical kind of reason to consume 1.2 grams of protein and not 0.8 grams or 0.6 grams or whatever. So you should be consuming 1 to 1. So if, sorry, if you were to take this, based on this, you should be consuming somewhere from 0.9 or 1 gram of protein to 1.2 grams of protein per kilogram of body weight per day. And if you consume that much, you don't really need to worry about any of this stuff. You don't have to worry. Also, you consume. So basically, you have to consume enough protein, and that protein has to come from more than one source. It doesn't mean that you get all your protein from just soy, or you get all your protein from just seitan or something like that. There has to be a combination of foods. And the way I like to do it is I just have a protein powder, which is a blend of pea protein and brown rice. And I consume rice on pretty much a daily basis. I also consume soy on pretty much a daily basis. I have soy milk in the morning and then tofu at night. And I also have rice, and I have protein powder. And all of these things are diverse enough to give me different DIAS scores. And as I mentioned here, DIAS scores are additive, which means that you can add the DIAS scores of different foods together such that you end up meeting your protein requirements, right? So as long as you're consuming enough protein and that protein is coming from diverse sources, you don't have to worry about protein bioavailability. You don't have to worry about digestibility. You don't have to worry about quality, etc. This is for vegans, obviously. So that's kind of the run through of the entire thing that I had to say. Now I can just if there are questions that are from that arise as a consequence of this, is well, you can just let me know in the comments and I'll take a look. R is the one that'll change. Yes, R is the alkyl group. Gelatin. Oh, it's gelatin, not collagen. Okay, fine. Lacks tryptophan. Um, Nitrogen. Why nitrogen? I don't know what that means. Like, what do you mean why nitrogen? The amino is NH3. Few amino acids have extra nitrogen, but roughly they are better for calculation compared to carbon. So nitrogen estimation by Geldol method is good for knowing the amount of amino acids. Um, you just crossed the line. I misspoke. You misspoke. I think you said PDCAS is better. No, yeah. If I misspoke, then PDCAS is not better. Diaz is supposed to be better. Um, yeah. Diaz relies on generalized nitrogen to protein conversion factor of 6.25 as standard, but plant proteins vary in nitrogen content, thus underestimating the protein amount. What do you mean by that? Um, what do you mean by generalized nitrogen to protein conversion factor of 6.25? Because, like, I'm not sure which part of the equation that you're referring to. Um, because the protein amount is going to be the same. Uh, yeah. I mean, yeah. That's pretty much, I think, everything that everyone needs to know. The idea is simple. Like, the take home message is that if you're consuming sufficient amounts of protein and the protein is coming from diverse enough sources, it doesn't have to be too diverse. Like, as long as you have more than like two or three sources of protein in your diet, you should be fine. So consume enough protein, consume enough consume the protein from more than two or three sources, and you should be fine. There's no need to be like um, spazzing out on how much protein that you're consuming. And yeah, so yeah, coming back, it doesn't really matter. Um, like, I'm actually kind of bearish about the whole ISKCON thing. When ISKCON is going to get rid of eggs and is going to just provide um, plant-based sources. Yeah, they're going to introduce some amount of dairy, but I'm not convinced that the harm from the introduction of some amount of paneer is going to be more than the amount of harm that is going to be from eggs, because you know um, yeah, like, eggs are just like um, you have to buy like way less, sort of, to drive like a signal and stuff like that. We'll talk about all that, but the idea is, I'm not convinced that the harm that's caused by the dairy, which is probably going to be there anyway, is more than the harm caused by the introduction of sorry, the harm prevented by the removal of eggs. So I'm actually um, I'm okay with this. Obviously, it's going to be better if the same amount of dairy is replaced with plant based. That'll be the best. But as long as we don't know if the eggs are, if the paneer is worse than the eggs, as long as there's no reason to think that, I'm actually okay with the whole thing, and people are losing their minds over nothing. The kids are going to be fine as long as they're getting enough protein. Um, and you, as a vegan, are going to be fine if you consume enough protein from enough diverse sources. Um, yeah, so I guess that's all I had to say. Let me know in the comments, even after the stream ends, if you have questions, and if there are enough things that need to be covered. I might consider doing a part two. But typically, most of the stuff should be clear by now. And if it's not, you can always join one of the Discord servers that I lurk in, and like just ask me, right? So yeah, y'all have a good night, at least for the people in India, and stay safe. And I'll see you maybe next week. So see you until then. Goodbye.